experiment, we will identify different types of plant tissues, parenchyma, sclerenchyma, and collenchyma with the help of prepared slides, and we will draw their labeled diagrams in the practical file. The material required for this experiment is permanent slides of parenchyma, sclerenchyma, and collenchyma tissues and a microscope. When you will see a permanent slide of parenchyma tissue under the microscope, you will see something like this. Let us understand what the structure is and how you will draw the diagram in your practical notebook. So, a transverse section looks like this, whereas a longitudinal section of parenchyma cells looks something like this. It is one of the simplest tissues present in plants, and the cells are basically isodiametric. So most of the cells will look like the same size and of the same shape. So isodiametric cells are there with relatively thin, unspecialized cell walls. No intercellular spaces are there at certain places, but at some places you might find some intercellular spaces. So you can say the intercellular spaces may or may not be present here. There's a prominent nucleus which you can see in the longitudinal section. The blue spot here is the nucleus that you can see. Cytoplasm has a big vacuole, so this is a vacuole that can be seen in the longitudinal section. And the main function of parenchyma is storage of food. So it basically serves to store the food for the plant. Sometimes it has some additional functions also, like in aquatic plants, it helps in the storage of air, which helps the plant to float, providing it buoyancy. Hence, it is known as aerenchyma. And sometimes it might contain chlorophyll also for photosynthesis, and in that case, it is known as chlorenchyma. So this parenchyma tissue is basically found in the epidermis, cortex, xylem, phloem, and the pith of the stem. This is the microscopic view of sclerenchyma fibers. So when you'll see a microscopic view of sclerenchyma, when your teacher will show the slide on sclerenchyma under a microscope, you will see something like this. Let us understand the structure of this now. Sclerenchyma fibers are always dead. So they are never found in the living state. It's basically a dead tissue. And the cells have very thick cell walls. So you can see in the transverse section that the cell walls are highly thickened because of the deposition of lignin. And there is no cytoplasm, no nucleus, no vacuole. Also, the cell walls of adjacent cells are connected with each other with the help of these pits. The gaps, these gaps that you can see here, these are known as the pits. The cells have very low water content and they are very like absolutely closely packed with no intercellular spaces at all. Also, the sclerenchyma cells are of two types, sclerenchyma sclerids and fibers. So you can see the difference. Sclerids are rounded in shape, whereas fibers, they are like kind of elliptical or spindle shaped in nature. The third type of tissue, collenchyma tissue, looks something like this when you see it under a microscope. Let us understand the structure and the key features of collenchyma tissue. Look at this figure. This will help you identify the key features of collenchyma tissues. The cells are polygonal in shape with irregular thickenings around the corners. So this is a special characteristic which is found only in collenchyma tissue. Also, the cells are living in nature. They have a nucleus and they also have a vacuole in the center. Cytoplasm is present and the intercellular spaces are highly reduced. This tissue is mainly responsible for providing flexibility to the plant, and for that reason it is found in the stalks of the leaves where a lot of bending and flexibility is required to prevent the leaves from falling off due to the wind, and also it's present in the stem to impart the strength to the plant so that it can withstand the strong currents of wind and other environmental factors.